two fellas here with us from the Purdue University Center for Cancer Research. Tim Bobillo will be our first speaker, and I want to just try to give him a good introduction here. He has led the development efforts at the Purdue Center for Cancer Research since 2006. He is a Purdue graduate with a degree in communication. He worked at WLFI TV 18, then in various communication sales positions before coming back to Purdue as Director of Development for the Purdue School of Aeronautical and Astronautical Engineering in 1999. Tim is married to the beautiful Mary Ann and has 4 children and 2 wonderful grandchildren, Bo and Della. So, Tim, I'm going to let you introduce Scott.
Thank you very much, Jim. It is a real pleasure for me to be here. I look out and I see some familiar faces, Emily among them, but thank you for inviting the Center for Cancer Research. Many of you may know about the Center for Cancer Research, but it's really important for us to share our message because we are leaders. Purdue is a leader in cancer research in the country. And that is not just me, Tim Bobillo, saying it because I love Purdue. The National Cancer Institute ranks cancer centers, and we are in that top group. Only 70 of the hundreds and hundreds of cancer centers have a designation by the National Cancer Institute, and we are one of those 70. And among the 70, we are rated very near the top. So they recognize the value of the research that is done at Purdue. We do not have a hospital, we don't treat patients, but our designation comes from our research. 7 of those 70 centers have their designation for basic research. Purdue, MIT, Jackson, Cold Springs Harbor, so there are 7 of those. Others have their designation for treatment, and some have a designation for both. Those are comprehensive cancer centers, such as Mayo Clinic, MD Anderson, Sloan Kettering. But Purdue can be real, you can all be real proud of Purdue and the leadership role that we have in cancer research. And I'm going to talk to you a little bit about what I do for the center, but then I'll introduce Dr. Scott Briggs to talk about research. As Jim mentioned, I've been at the Center for Cancer Research since 2006. It's my job to raise money for cancer research, but also for communication and events and marketing, and so we have a little staff that does that. But yeah, it's really my role to be out and talk to people and to share our mission, and hopefully that matches up with their interest in cancer research, so that we can have more resources. The more resources we have, the more advances we can make in cancer research. Purdue has, the center consists of about, right now, 120 researchers from all over campus. So they're in biology and chemistry, as any research entity would have, but also Purdue is a real unique environment to look at cancer research because of the breadth and depth of our researchers. We have researchers from engineering who are looking at how to deliver drugs to the cancer. We have researchers in pharmacy on how to make drugs. We have researchers in foods and nutrition on prevention. We have access to the School of Veterinary Medicine, so we can look at animal models. It turns out that some canine cancers are very, very similar to human cancers. So, looking at canine bladder cancer, it gives us a real advantage as we're moving drugs forward. Those 120 cancer researchers, as I said, our charge from the National Cancer Institute is basic research. How does cancer start? Why does this cell go crazy? And so researchers like Scott are looking at models on how to find out the answers to those questions. But then also we're working in what's called translational research. How do we take those discoveries from the table to the bedside? How do we get those into patients? But whatever the cure for cancer is, it's going to start with research. And so that's where the Center for Cancer Research comes in. As I said, we're trying to get the message out to as many people as possible, trying to raise money. Somebody asked me recently, what's the largest gift you've ever gotten? And the individual gift is $2 million, but real quickly, Just shortly after I started, I went up to see a couple who had been, they'd been approached a number of times about making a large donation to the center. And I had been with them often, but they kept deflecting the conversation. So finally, I was going to go up and I was going to make sure I got an answer. So we went to lunch. He had invited a lot of people, so I couldn't have the conversation then. We go back to his office. It was just the gentleman and his wife and me. And so I brought the proposal out once again. And to my surprise, he said, okay, we're going to do that. What do you do next? Well, I was just flabbergasted. And I knew, having been in fundraising, that even though they agree, it takes sometimes days and weeks and months to actually get it finalized. Just because of paperwork and agreements and attorneys and financial advisors. And so he said, so what do we do next? And so I looked at him and I said, well, it would really be great if you just gave me a check for $1.5 million. Just kind of as a joke. His wife pulled out her checkbook and wrote a check for $1.5 million. So that hasn't worked every other time. There is a little information on the back. I'd like to thank you if you wear those wristbands around and just support the Center for Cancer Research. And again, locally, you can be really proud of what Purdue University is doing in cancer research. Next week, certainly any of you who are Purdue fans know that is Purdue Day of Giving, an effort where we really try to raise as much money in one day as we can. And so there are a lot of ways you can give, but I'd like to encourage you to make gifts. The Center for Cancer Research has a match by the Walther Cancer Foundation. That any money given to an endowment at the center is matched. So if you gave $100 to an endowment at Purdue, at the Center for Cancer Research, that would turn into $200. So that's a great way to do it. And I want to also talk about something that you may have heard about. Tyler Trent, a young man who became nationally famous. I got to know him. He was very close to the Center for Cancer Research. We actually appointed him to be a member of our advancement board. Last fall was a remarkable time because he just willed himself through the faith of God and his family to live out his dreams. And so a lot of opportunities were presented to him. And he took on the Center for Cancer Research as one of those entities that he supported. So we have started a fund at the Center for Cancer Research, the Tyler Trent Cancer Research Endowment. And that money, because it's an endowment, is matched by Walther. And so just last November we started that, so less than 6 months, we've raised $660,000. And that money is doubled, so we now have $1.3 million in the Tyler Trent Fund. So that is a great way to support cancer research. That was one of the most important things in my professional career, I think, is getting to work with Tyler Trent.
Yeah.
And just one final note, I was just informed that Noble Kaiser, who many of you knew, he had asked for memorial gifts to be sent to the cancer research. So those are always really important testimonials to somebody's recognition of the work we're doing. So I appreciate that very much. But on a personal note, I live in the house that Noble Kaiser used to live in on Ravinia in West Lafayette. So yeah, I had an opportunity to meet him a couple times in town. And he was just a charming man. So now the reason you're here is not to hear about me and my story, but Dr. Scott Briggs has been at Purdue for 13 years. He is a native of Iowa, grew up on a farm, went to school in Iowa, and then grad school in Nebraska. He is a professor in biochemistry in the College of Agriculture at Purdue, and he works on basic cancer research, and is also a great teacher. He is a father of 3, and coming from a small town in Iowa to West Lafayette, he is a real important member of the center. Plays a leadership role in some of our administrative efforts at the center. But his discovery, as I said, sometimes, a lot of times, these aren't huge announcements to be made about progress, but researchers, just continue to go after every next step. And science, many of you may be scientists or researchers, but it's a really important part of what goes on at Purdue University. And so I would like to introduce Dr. Scott Briggs.
All right.
Well, first of all, thank you for inviting me here to give a talk. Okay, sorry. All right, well, first of all, thank you for inviting me here to give a talk. Like Tim said, I'm actually a farm boy from Iowa. I came from a small town. It's, most people say they graduated from a small high school of 100 or 200.
Right.
Well, graduating class, my graduating class was 23. So it's a small town, about 500 people. So I always usually beat everybody when they say they graduated from a small town. Then I did my undergraduate work at the University of Northern Iowa, and then that's where I got interested into doing biological studies and research. I did an summer internship at the University of Nebraska Medical Center at the Epley Cancer Institute. There I worked for a while and then became a graduate student and graduated there. Sorry. And then I moved off to do my postdoctorate work at the University of Virginia in Charlottesville. It's a beautiful area, beautiful campus at the medical school. And then I got my job here in 2003 at Purdue. So today I am going to be, one other event that I should mention about supporting cancer research is the challenge event, which is the 5K run and walk. So that's always a good event because all that money goes back to us researchers, and I was one of those recipients to get some of that money. Okay, so today I'm going to talk to you about some of the research that I've been working on, and I'm not going to show you a lot of data that we're showing, but just more general concepts of what we're trying to do. And I don't think it works in this regard. So today I'm going to talk to you about hypoxic growth, drug resistance, which are couple characteristics of cancer that prevents us from killing cancer. And I'll be using this in a model organism. A model organism is a non-human organism that we use to study to try to put insights into how cancer cells grow.
It's changing here.
Maybe exit out again.
I'm sorry.
Okay. So again, I'm going to show you a brief information about the Cancer Center and the funding. So most of the administrative staff is housed in the Hanson Life Science Research Building, and they're housed on the main floor, and then above that, on the second, third, and fourth floor are cancer researchers, and I'm actually housed on the second floor. We have, obviously everybody can't be in that Hanson Life Science Building because we have over 118 scientists that do cancer research, and representing over 19 university departments. And we also have various new buildings that have been built to help support cancer research. We have the Bentley Multidisciplinary Cancer Research Facility and a recent drug discovery research facility as well. Like Tim was saying, we are an NCI-supported cancer center. This is a support from the NCI, which we get funds to help support the salaries of the administrative staff, but also the core shared facilities that us researchers need to do our research with. And as Tim was indicating, we are one of the 69 NCI designated cancer centers. There's over 1,500 cancer centers in the nation, and we're one of those. Of those 1,569 are NCI designated. That means they get money from NCI. And we are also only one of the 7 basic cancer centers. And we also have had this support for 40 years now.
Could you tell us again what NCI means?
National Cancer Institute, sorry. Okay. So, in the Cancer Center, the PCCR as in abbreviation, is 4 research programs: the Cell Identity and Signaling Program, the Chemical and Structural Biology Program, and the Drug Delivery and Molecular Sensing Program, and the Medicinal Chemistry. We have various groups of faculty members that have various interests grouped in these different programs. We also have focus groups on various different cancers, such as bladder cancer, brain cancer, breast cancer, obesity and cancer, prostate cancer, and pancreatic cancer as well. And I'm part of the Cell Identity and Signaling Program. This includes 33 members across campus in 7 departments and 5 different colleges. This program focuses on a lot of cell biology and the various organisms or model organisms, such as people using mice, zebrafish, human cell lines, Drosophila, as well as yeast. And this is what organism I study. And then other people study various molecules in this programs that oncogenes that lead to or develop cancers, and then also how genes are being turned on or expressed. Okay, so there are some various causative and contributing factors of cancer. So most of us probably know that various mutations in our DNA can lead to cancers, such as mutations in oncogenes, which if overexpressed can lead to uncontrolled cell growth. Or we have deletions of certain genes where you delete them, and then you can also lead to overgrowth as well. There are also various contributing factors for cancer. We have drug resistance, and hypoxia is one of them. An area that I study is epigenetics. This is various environmental factors that can help contribute to cancer. But today I'm going to talk to you about hypoxia and drug resistance. So hypoxia commonly occurs in tumors. So a hypoxia environment is deprived of oxygen. So some examples of oxygen levels around normal atmospheric oxygen that we're breathing is around 20%. Physiological tissues inside our bodies are around 5 to 10% oxygen, but tumors are less than 2% oxygen. And when we have very little oxygen around tumors, it can lead to drug resistance, where they're resistant to radiation and chemotherapy treatment. It can lead to metastasis, altered metabolism, so changes in the tumor that we can't then treat, and also changes in gene expression. So certain factors that get expressed to make them lead to all these other types of features. So here's an example of a tumor here, and the reason why it's hypoxic, so here's a blood vessel. This region right here would be what we call normoxic, or containing oxygen, and the further away we get from the bloodstream, it becomes hypoxic, and that's detrimental for us. Okay, so now that's the cancer, but there are other organisms that actually can naturally survive under hypoxic or anaerobic growth. Anaerobic growth would be 0 oxygen, and that's bacteria and fungi. And I focus on the fungi yeast aspect of it. And that we're hoping that will also inform us of how we can actually treat these hypoxic cancers. Okay. So we use model organisms. The organism I study is Saccharomyces cerevisiae. I don't know if you've ever heard of that, but you probably used it. It's actually called baker's yeast. It's yeast that you use to bake with. So we use that as a system to study how cells grow. So here, human and yeast cells, they're obviously evolutionarily distinct. So here's a Saccharomyces, what Saccharomyces look like, and a human dividing cell. This is a dividing Saccharomyces cell. they have obviously similar cellular structures, although there are some distinct differences. But the interesting aspect of it is that they share very similar genes with similar functions. So some of the genes we could study in Saccharomyces will all behave the same way as in human cells. The reason why we study these yeast cells is we can do genetics, we can do gene deletions in them, we can do biochemistry. There's been major discoveries from Saccharomyces. 11 Nobel Prizes have been discovered using this organisms. You could do drug testing, and it's translatable. Like I said, various genes in here contribute to what happens in human cells. So what I study is the area of the cell, is the DNA inside the nucleus of a cell. And this is a blown out version. So we have our genomic information stored in the nucleus. There's about 2 meters of DNA, which is about 6 feet of DNA in our nucleus. So it has to spool around these proteins called histones in order to fit inside the nucleus of a cell. It has to be really compact. And I study how genes are turned on and off in that cell, and try to understand what genes are contributing to cell growth. All right, so here's one of 2 pieces of data that I'm going to show you. Okay, so we're interested in identifying genes required for hypoxic growth. So what you see here is, over here, a plate of yeast grown on a semi-solid media. It's kind of like Jell-O, but a little bit stiffer. And where we put yeast at different concentrations, from high concentration to a low concentration here. And this plate here has been grown under aerobic conditions, where just like ambient aerobic conditions. The WT is a wild-type yeast with no mutations. And the rest of these, you don't have to worry about the names of these, but these are various deletions that we created in yeast. And as you can see, that when we, under aerobic conditions, if we knock out these various genes, we don't really see any growth difference compared to that of wild type. So it's not really having any impact under aerobic conditions. But if we take that yeast and grow them under hypoxic conditions, we can actually, you can probably see some differences going on here. So wild type yeast still grows like it should because it can adapt to that environment. And then when we start making out single deletions, double deletions, or even triple deletions, we get a worse growth phenotype. So they can't grow as well. So because there are similar genes in humans, we're hoping that if you knocked out these genes in humans, or probably not knocking out, but you'll probably develop a drug to these genes, could you inhibit potentially hypoxic growth of the tumors? And the good thing is that you probably wouldn't affect just normal tissues because they're growing under aerobic conditions. Okay. So the other causative cause for cancer is drug resistance. We can't kill the cell because they become resistant to drug treatment. All right. So the interesting thing about cancer, human cancers, and yeast or fungi, they have developed a very similar drug-resistant mechanism. So some of the drug-resistant mechanisms are decreased uptake in drugs, so the drugs won't be taken up in the cell. You can also have increased efflux. So we have these drug pumps that pump out drugs. You increase those drug pumps, and then you're pumping out, you're treating the cell, but the drug's going in, but then you're pumping it right back out faster than you can give it to them. We have changes in metabolism. We have altered drug targets. So the drug target that we have gets mutated, let's say. Now it's not effective anymore. Or we have resistance to cell death. Now, we usually want to obviously kill the cell, but now they can't die. And a lot of these are due to changes in, again, gene expression, how genes are turned on or off. Okay, so now I'm going to give you some insight about some fungal aspects. So fungal drug resistance is on the rise, mainly because of use of antibiotics and antifungals. There are some newly emerging fungal pathogens. So Candida auris, I don't know if you've heard, that's been in the news recently, about a highly resistant yeast, a fungal strain that has really just recently been identified in the last few years. And as actually has increased in the presence in the United States recently as well. So some of the newer cases have been out in Chicago. And we haven't actually really developed any new antifungal drugs since around 2006. Right. So here are some of the major fungal pathogens. There's Aspergillus, Cryptococcus, Candida albicans, Candida glabrata. And glabrata is the fungal pathogen that we study as well. So fungal pathogens are a major concern, global concern, because over 1 billion people suffer from fungal infections. These aren't necessarily all fatal. Could be more just on the surface of your skin. So athlete's feet, ringworm is another just surface fungal surface on our skin. But if it goes systemic, it can lead to a lot of deaths. And about 1.5 million deaths occur from fungal infections. And we also have an increasing population of immunocompromised patients, which are then become more susceptible to fungal infections. And if you have a systemic fungal infection, you have probably a less than 50% chance of survival. And because these fungus and human cells are very similar, they're hard to treat, and because they're becoming drug resistant, they're very expensive to treat as well. So it's costing us billions of dollars in healthcare costs. Okay. So some of the risk factors for developing systemic fungal infections are cancer chemotherapy. So great, we're going to treat you with the chemotherapy, but the one risk factor is that you're suppressing the immune system. Then you can pick up a fungal infection or a bacterial infection, which of course we'll have to make sure we need to be developing these treatments as well. And other disorders that suppress the immune system, if you have HIV, which suppresses the immune system, burns, diabetes, and of course if you have cancers in the blood cancers, you're going to have a suppressed immune system as well. Okay. So there's different types of drug treatments for fungal infections. We have the azole drugs that target the ERG-11, which is involved in producing ergosterol, and the ergosterol is the cholesterol of yeast. So we have Paulines, amphotericin B, which targets actually the ergosterol or the cholesterol of yeast. We have echinocandins, which target the cell wall of the yeast, which is unique to yeast and not to humans. But the idea that we want to do is, can we identify potentially new drug targets? So again, here's the same type of assay that I showed you before, is when we grow wild type under normal aerobic conditions, it grows, we knock out this gene, and it grows very similar. However, if we treat with the azole drug now, wild type grows better than our gene deletion here. So showing that wild type is more resistant than our gene deletion. So hopefully, potentially this protein might be a potential target for a new drug, fungal drug. It could also be potentially used as an anti-cancer drug too, because the human form of this SET1 is also involved in developing cancers. Okay, so hopefully today I helped to show that the use of these model organisms to help us to understand how various aspects of these contributors to cancers, that hopefully we can identify genes that disrupt these organisms' growth. Potentially we could do the same thing in cancer as well. Okay, so lots of people to thank. I'd like to thank my lab up there. These are former past graduate students and undergraduate students that I've had in my lab. And of course, funding from the NIH, National Institute of Health, Purdue Cancer Center, as well as the Challenge Research Award as well. And as well as various parts of Purdue and the Department of Biochemistry and the Sidney Kimmel Foundation for their funding as well. with that, I would take any questions.
I have one over here. The ultimate research that you're looking for, is it a pill, something that a person can take in order to eliminate the cancer?
Yeah, so it'll be either a pill or something that has to be injected IV by IV. Yeah. But
How far away are we from
from
a pill?
from a pill? That could, from the work that we're doing right now to then, there could be a many years, because we've established our evidence in a model organism. We would then have to go then try it on human cell lines, and then go into more of a animal model, and then go into clinical trials. So many years but we have at the Cancer Center, we have people in various stages of that.
Yeah.
We have people doing clinical trials right now, but it has to start with the findings first, then that can eventually lead to that discovery.
Is there anything particular that you're doing for pancreatic cancer?
So I'm particularly not doing anything, but I was on a committee that just recently hired a new person to study pancreatic cancer. So there's been, so we're very interested in studying that cancer.
Is cancer hereditary? Can you pass it on, if you have it, can you pass it on to your kids?
It depends on where in the body it is, yes. So if it's germline, if it's in your, then you can pass it on to your potential kids. If it's just somatic, then it won't get passed on. So somatic is that the germline would be in your like sperm and ovaries. There have to be a mutation there in order to pass it on. If you got somatic after you were born, those cancers won't be inherited.
So the National Cancer Institute is strictly for the United States. My question would be, as a researcher, how do you coordinate with other researchers and keep up to date in terms of what other places or centers are doing so that you're not duplicating necessarily and spending time on duplication, or that you're taking someone else's findings and branching out from there, or they take your findings and go from there? How do you coordinate?
So a lot of times that there is coordination. We talk to interact with other people in the field that we have like interests in. But there are things where, like for my work, I'd be publishing it, and hopefully somebody else would then see that and go, oh, maybe I can go start studying in human cells, and then doing that work to advance it. So I'd be in this, and hopefully spreading out the news when I publish that, then somebody else would take that observation and maybe carry it further as well.
So is there a centralized database that the people, let's say in other countries around the world, can tap into and see your findings?
Yeah, so we publish them in particular journals, and we have access to those journals through this database. One of the databases is called PubMed, where people publish, and you can go click on that and get all their information. Yes. And then if you really want to collaborate with them, the scientific community is very collaborative. If you want to collaborate with them, you just call them or email them and start approaching them. Or we also have conferences as well that we go then present our data and information, and then that's a very good way of doing that as well.
One more question back there.
How lengthy is this process from the time you make a discovery in your laboratory to the time it ends up in the form that a doctor can prescribe?
That could be like, you're talking about, it could be many years from very initial discovery to going on to, I mean, you're talking about, it could be up to 15, 20, 30 years, but it could be shorter sometimes too. It depends on how well that discovery works. So it could go very well and then get stopped for a particular problem. And then, let's say it's too toxic, let's say, for the rest of the body. So then you might have to go back and reformulate something again, to hopefully it's not toxic for the rest of the body.
So it goes from your lab to somebody else taking does clinical studies?
Yeah, so I wouldn't do any clinical studies because I wouldn't have the expertise. I would have to either collaborate with somebody or they would be interested in doing that, yes.
So real quickly, here's a real world example of that. So many of you know about Phil Lau.
Right,
He's
right.
a researcher here. He started Endocyte.
Right.
And when I first started at the Center for Cancer Research, I knew of Phil Lau because I've been around Purdue. And so I was going around to meet some of the researchers, and I sat with him in his office, and he was showing me some examples of some of the work he was doing. And I asked him, how long have you been working on this? And this was in 2006. He said, 19 years. So that was 13 years ago. So now, what is that? 31 years later, they've sold that drug, and it's almost, or not the drug, but the molecule, and it's been used in clinical trials, but it very shortly will be used in patients. But that's 31 years of work that he's done to get to that point. Now, not all of them take that long, and some of them never reach that far at all, and some of them take longer, but it's very long, laborious, and a very expensive proposition to study cancer research and to come up with cures, but it's worth it. And so, yeah, I just want to encourage you to.
But this is why we need the basic research, because we need to find more targets. We have more targets. Hopefully, if you only have one target and if it fails, then that's it, right? If you have 10 targets or 30 targets, that can be hopefully eventually translated through. If 3 of them make it through, that's better than 0. So that's why we need to do this basic research to see how cells grow, so that we can identify more targets, so that we can hopefully have more successes.
Thanks.